So it is really a pleasure to introduce Michael Chapman today from the Department of Biochemistry at the University of Missouri. Michael was actually my graduate mentor, and it's with him that I kind of got my feet wet with modeling. Today, he's going to tell us a little bit about his efforts in optimizing consistency between model and experiment. Michael, take it away.
Well, thank you very much, Omar, and it's indeed a pleasure. And thank you so much for organizing such a great workshop. So I'm going to dive right in, and I'm going to start by asking some very basic questions. And I'm going to proceed slowly. Oh, I need to share screen, right? I'm sorry.
Yep.
Uh, is that looking good for you?
Yep, looks good.
Okay, so I'm going to proceed slowly, asking some questions, and slowly because I want you to be thinking a little bit about the questions and what you might be trying to do. Why are we optimizing? And I would say that we're trying to improve the accuracy of our models and to jiggle the atoms a few tenths of an angstrom. Sorry, I'm not seeing the slides getting updated. There we go. Jiggle the atoms around a few tenths of an angstrom. Little movements that might be critical in discerning functional interactions, such as hydrogen bonds. What should we be optimizing? I think in vague terms, we can all agree that the model should best agree with an experiment. And I just give brief mention to something that Pavel talked about, that with electron microscopy, we're usually not at resolutions where we can refine independent isolated atoms. And so we'll have to refine with supplementary restraints or constraints for ideal stereochemistry. For this talk, I'm going to take that as a given, and I'm not really going to say anything more about it. But what do we mean by improving the consistency of model with experiment? Exactly. And there are several possible answers. We could place atoms at the highest density. And I note here that I'm talking as a crystallographer, as if this is electron density, but you know that really what we're talking about here is a Coulombic potential map. We can fit atoms within an isocontour, within an envelope, a bit like we all do using software like Qt. And we can get progressively more sophisticated, trying to maximize the agreement of density values between calculated and observed maps at points in the map. We have further decisions to make as to whether we're going to do this evaluation just at atom centers or whether we're going to use all map grid points. Now, each of these choices in mathematical jargon in terms of objective functions or targets to be optimized. And there are programs that encode every one of these. And my goal today is to talk about some of the pros and cons of each and the circumstances where the differences might be important. There can be principled reasons for the choice of objective functions, but pragmatics and history are also playing large parts in the current standard operating practices. So I start with a little bit of history in that structure refinement in electron microscopy has a heritage that descends from crystallographic refinement and also from molecular mechanics simulation. Crystallographic refinement of proteins really descends from the work of Lyle Jensen and colleagues, who were the first to demonstrate reciprocal spaced refinement of proteins, where you're optimizing the agreement between observed and calculated structure amplitudes, where what we note here and what they noted is that the Fourier amplitudes here are untethered from poor experimental phases. And they were also among the first to show the value of supplementary stereochemical restraints. Now, what is now mostly forgotten in our community is that this work was preceded by real space refinement that had been encoded by Bob Diamond. In this type of refinement, you're minimizing the difference between calculated electron density and that of an observed map. Bob Diamond calculated this density here from spherical Gaussian functions placed at every atom center. And similar implementations have followed in Frodo, O, Coot, and it was Bob Diamond's work that was the inspiration for what we did in my lab. So I want to turn your attention back, a couple of decades to thinking about the needs of structural biology about 25 years ago, when EM structures, you were doing well to get resolutions between about 1 and 3 nanometers, and people started fitting crystallographic component structures into these maps. And soon there was a demand for methods to fit these structures, first by rigid domain, and then in about 2005, people started working on flexible fitting methods, and that we started to see progress towards much higher resolutions in about 2013. And throughout this time period, there was a lot of motivation to be using the methods from crystallography adapted to electron microscopy. At that time, we didn't anticipate the growth in electron microscopy and the extension to higher and higher resolutions. And so the motivation was to adapt existing tools rather than to develop new ones. Now, Bob Diamond had demonstrated a perfectly good algorithm, and why is nobody using it at this point? At the heart of his method, Bob Diamond was calculating the density contribution of each atom as a spherical Gaussian, and I think Pavel showed the formula earlier. This is with the assumptions that the data of infinitely high resolution are not attenuated with resolution. These are both unrealistic assumptions, but Diamond argued that the image of an atom involved the convolution product of several transfer functions that together could be approximated by a Gaussian of increased width. This works at high resolution, but would require ad hoc calibration at lower resolutions where the approximation deteriorates. In addition, there were several practical considerations. Diamond's program was all-inclusive in terms of stereochemistry and optimizer, model parameterization, all of which are now dated. And we really needed a module for just the real space target function that could be combined with more modern modules for other functionality. Finally, by about 20 years ago, many were wanting to use molecular dynamics methods for flexible fitting, and anticipating that Diamond's algorithm would be too slow for the many functional evaluations that are needed in such algorithms. So the the question of speed comes up because for each grid point, you've got to sum the contributions of neighboring atoms from several neighboring atoms and have to do this many times over. And so we've heard some talk already about what one needs to do in terms of resolution-dependent refinement. And there are a couple of ways that one can do this. And for instance, the program RefMac transforms everything into reciprocal space, both the observed map and also Fourier transforms the model, and the optimization is done in reciprocal space. Now, many have argued that this is an exact equivalent of what we would do in real space, and I contend that it's actually only an approximation. And the way that people argue that it's an exact equivalent is through Parseval's theorem. But Parseval's theorem only really applies if the Fourier integration volumes are identical. And so the global reciprocal space refinement of an entire structure is not necessarily equivalent to real space refinement of subregions. Overfitting can be exacerbated in reciprocal space because of compensating errors. Two wrongs in different parts of the molecule can collude to improve the fit to the overall structure amplitudes. Now, this is mitigated in RefMac, where structure factors in the complex plane are used, and this thereby brings in the phase information. A second issue is that electron microscopy structures tend to be large and sometimes symmetrical. And so there can be a temptation of expediency to cut out a region of interest, a unique region of interest, and refine just that sub-area, masking out the rest of the map. Care is needed in doing this to make sure that you don't introduce any Fourier ripple errors due to the sharp boundaries that can occur at the edge of a mask. So we approach this resolution dependency in a different way. And the other way that you can do this is to calculate the expected density from each atom by Fourier transform of the relevant atomic scattering factor. On the left, we've got the scattering plotted as a function of reciprocal distance. And on the right, we have its Fourier transform, which is strength of density versus distance from the atomic center. This approach is perfectly general, and it can be used to calculate the expected map for X-ray, neutron diffraction, or electron microscopy, as long as you're using the appropriate scattering factors. The scattering factors are all tabulated for all of the atoms that we're likely to encounter, with values that have been obtained either by experiment or by quantum mechanical calculation. And then they're frequently approximated by a sum of Gaussian functions. In our implementation, we discretize the scattering curve. I'm illustrating this in one dimension, but in reality, we're using three-dimensional functions to obtain the density in a volume surrounding an atom. And so this here schematically illustrates that when we go from one dimension here, looking like a histogram plot, we go to shells of different scattering profile in 3D space. And the advantage in doing this is that there is a closed form Fourier integral for this shell here, and it takes the form of a half-order Bessel function. Now, this looks like it might be expensive to compute, and so it is. And more on this later. another couple of things that I'd like to note. We're assuming that the scattering falls off with spherical symmetry. We haven't yet incorporated anything for an isotropic thermal motion. Now, from the closed form integral that we've got here, what we are essentially doing is taking a shell for each shell in reciprocal space through this formula here, calculating its Fourier transform, and then adding the components to get this scattering, to get this density function, atomic density function in real space. This is essentially perfectly accurate. If we go to the limit of infinitely thin resolution shells, we get an exact replication of what the atomic density function should look like. Now let's think about what happens if the scattering is attenuated, and in its most extreme case by a sharp cutoff in resolution that we see here, that we've already seen this morning, that the effect in real space is that you then see a ripple effect extending away from the atom. And our way of calculating the atomic density takes this into account perfectly. Here we see some examples for atomic density functions calculated between 2 and 10 angstrom resolution. You can see that the atomic density function gets broader at low resolution. Calculating the density for each individual atom is only halfway there. And there are a number of objective functions that we can use in atomic refinement. And the one that we're using is a least squares objective function, where we're minimizing the squared difference between calculated and observed density at a set of grid points in a volume, and we have to scale. So there's a gain term lambda and an offset level S. You can do something very similar with correlation coefficients, and both of these are pretty expensive computationally, and even more so when you're calculating the density by Fourier transform of scattering factors. When optimizing by gradient descent, there is some offset because we have available analytical partial derivatives from the closed form integrals. And so we get the derivatives that we need for optimization with very little extra calculation. Now, when you're doing molecular dynamics optimizations, there's no gain from that because you're entirely using a set of objective function evaluations at a lot of successive points. And so this really drove the motivation to find more expedient approximations. And there are many of these that work. Now, here we're going to be looking at both least squares residuals, where we see a minimum at the true structure. So what I'm plotting here is the objective function value with error along the horizontal axis, as we're taking, in this case, a peptide structure and translating it by an error that is listed here. We also have correlation coefficients that have a maximum at the true structure, unless we just flip the sign. These are correlation-based objective functions here with the sign flipped, so that the minimum is at the correct structure, which allows one to do a to use library routines for minimization. Notice that the correlation coefficients have slightly wider convergence radius, but least squares is a bit more sensitive as you get close to the convergence. But viewed this way, both look like they will work. So let's look at some approximations now. In this first one, you can see that this is very close to being a correlation coefficient, providing that we normalize our maps, that we have each of them with a mean value of 0, so that we get rid of this term here and this term here. And we also set the standard deviations of both of our maps to 1. And then we end up with this simplified function here. It is a correlation coefficient, but it now becomes apparent that this is an overlap integral. It's just the product of two fields. And so that's already a little bit quicker. It becomes a lot quicker if instead of using the map at all grid points, we now use just the map at all atomic centers. Now, this makes for a pretty fast approximation, and you can go even further. And this is the fast method that's used in Phoenix Real Space Refine, where you now replace the calculated density at each atomic center with a weight that's really just proportional to the number of electrons or the atomic number of each atom. And so this then becomes a very fast evaluation and is attractive for many purposes. The last two lines here embody heuristics that I don't see as being used in recent years, and so I'm not going to talk about them today. So now we're going to take a bit of a closer look, in that the curves that I was showing previously are the average of many different evaluations where we're introducing a displacement, an error in a structure, but we're moving it in random directions. And the smooth curves are the averages of many evaluations in different directions. Now, the challenge is what's happening down here. So this is our fastest approximation. And what we find is that a few tenths of an angstrom away from the correct structure, among the many different directions that one could go, you're finding some that are giving a lower objective function. And so the average value here is, what we would expect, but we've got a diversity of scores depending upon exactly which direction we move. And a good optimizer is going to find the lowest value. For the example that we've got here, it would put the structure 0.4 angstrom away from the correct structure with a 3 angstrom map. Now, this is, we don't actually see this when we do refinements in practice. And why not? It's because usually we've got additional stereochemical restraints that are also in play. And so if we move a group of atoms to a place that's not exactly where they should be, we're going to have geometry fighting that. And so what we end up with is something that's more restrained. But in reality, what's going on is we're getting a little bit of fighting between a good fit to the map and good stereochemistry. And this is meaning that we're not at the very best structure that we could be at. So the rub against our method was that it took too long. That's, I believe, no longer the situation. For a typical protein complex, it takes about 5 minutes to do a batch of gradient descent minimization, about an hour for simulated annealing. And these are times that are measured on a 6-year-old workstation that then cost about $5,000. Now, these are slower than competing algorithms, but they're now, to my mind, perfectly acceptable. And this is not what it used to be. It's much improved. And the improvement is due both to faster computer hardware, but it's also resulted from optimization of our code. And so what we've done are some of the easy things in that we've vectorized the algorithm the easy way by using NumPy array arithmetic so that our code remains in pure Python. We're also caching expensive parts of the calculation that are repeated many times. And so we just save for future use the densities that we've calculated as a function of distance by atom type and for similar B factors. And we're also caching the partial derivatives that we use for optimization. There's a lot more optimization that we could but haven't done. And conceptually, this is an algorithm that would be very parallelizable. We've got atoms that we could do calculations on in parallel. But with pure Python, the global interpreter lock would mean that we would actually see very little gain from doing this unless we were prepared to reformat this with a C extension. We also haven't yet tried to exploit what we could do with graphics cards. We haven't experimented with CUDA. However, I think that the code is fast enough that unless it gets really very broad usage, it's not justified in spending the time for further optimization. So what does our code do? We compare calculated and observed density, where the calculated density is coming from Fourier transform as scattering factors. We compute partial derivatives that we're then using for optimization of atomic positions, isotropic B factors, occupancies that we can also do gradient descent optimization of magnification as we can for various imaging, electron microscope imaging factors, including the envelope function. We can also refine a signal attenuation function and a resolution limit. We'll talk about that more later. We've got syntax available if you want to do refinement of rigid groups or to group B factors, occupancies, etc. And we provide access to standard optimizers for use in a standalone mode. But I would emphasize that we are not supporting stereochemical restraints. I don't want to reinvent the wheel, and I would never have the resources to support such an extensive commitment. So we're relying on external programs for the stereochemical restraints. And until recently, probably the best of these is, or most appropriate has been CNS, and into which we embedded an interface to our module. But this is really now at the stage of being considered legacy code. I think there are a few electron microscopists that are familiar with the CNS user interface. The code is no longer supported. It gets increasingly challenging to compile, and it's more and more environment dependent. And so we've made the decision to deprecate our CNS interface. So we're in a transition, and I think we're well on the way, but not done yet, with providing a path to pip installation. And so this is a standard Python way of installing a module and all of its dependencies. And in this mode, our module would be available from a public software repository. Now, this requires that the license be open source, or at least copyleft. And this was a bit easier said than done, because my former institution, OHSU, held the rights to code developed for an earlier version. And so in May, we received permission to proceed, subject to removal of some deprecated code that's no longer needed. Now, I have to indulge your patience in some of this because my day job is as a chair, and so my first responsibilities are to 25 faculty and their research and teaching missions. And then my second responsibility is to our funded research program, which is on the structure of gene therapy vectors. And so really the development that we're talking about here has to be a hobby. And there are long periods where I don't get time to work on it. But in the future, we envision that we will provide a module that will encourage other people to access it through their own refinement programs. Now, there's a very small history of doing something similar. The DEN program that was developed in the Brunger and Levitt laboratories a while back used our algorithm, but I think that they coded it independently. I've spoken about the refinement of imaging parameters, and the most important is an effective resolution limit. What we're doing is applying a Butterworth low-pass filter in the calculation of our model map density, refining its parameters for best agreement between the observed and calculated maps. We're choosing a Butterworth filter because of similar functional form to typical Fourier shell correlation coefficients. And our method works with both original and with sharpened maps. The functional form is over here. There's a cutoff frequency or resolution, and Henderson gave us the method by which we can reconstrue this as the FSC 0.143. We can calculate directly from the omega c, or what should correspond to the FSC 0.143. Now, all of these, so we refine an attenuation, we can refine an envelope function or a B factor, and all of these plus atomic B factors are all incorporated within this contrast transfer function here. And this is closed form. All of its parameters are refinable, but we do not recommend that you try to refine an envelope function at the same time as either the filter or the individual B factors, because they're so covariant. And in addition, we're also refining a magnification factor. So for the balance of the talk, I'm going to give some illustrative examples of how we've used refinement, examples from the Chapman laboratory. And just one slide of biomedical introduction, that we work on adeno-associated virus, which is a small single-stranded DNA virus that is now the leading vector for gene therapy. And in gene therapy applications, you are replacing the infectious DNA with the transgene that you want to convey to cells. It has now been approved by the Food and Drug Administration for 7 different treatments, of which the most notable, spinal muscular atrophy. This is a debilitating disease with a life expectancy of about 2 years. And now the first children from clinical trials are about 7 years into post-treatment. And many of them have now a fully normal life, as far as we can tell. And so this has had tremendous impact on those families. And then more recently, single dose treatments for both hemophilia A and hemophilia B have become available. Now, there are a number of challenges, and indeed 7 patients have died in clinical trials due to immune-mediated liver toxicity. And the reasons are that AAV is not very efficient. The biggest problem is that it takes thousands of virions to guarantee that you'll get a single copy of DNA into the nucleus of a cell. And then also, we've got to remember that this is a virus, that our immune systems are targeting the vector unless we've got immune suppression. And so combine these together, and we're using very large doses. And we hope to learn enough about the fundamental structural biology to be a foundation for the design of more efficient vectors. I'm going to run you through several examples of EM structures and their refinements in chronological order. And so we start by going back 14 years to a nanometer level structure. But I note that still many EM structures are in mid to low resolution domains, and it's essential that our atomic refinement methods work well for those cases. For this example, the initial structure was modeled using the known crystal structure for AAV2 and a homology model for the FAB fragment of an antibody that was bound. The virus atoms were fixed, and the statistics that I'm reporting here are those just for the FAB over here. When refined from a manually fit model, we saw a 10-degree rotation and improvement in correlation coefficient from 0.68 to 0.78. We had a refined magnification change of nearly 2%. I would note that now the calibration of microscopes is better, and we're seeing smaller changes to the magnification. We then treated as separate rigid groups, the variable and constant regions, and there was a further improvement to a correlation coefficient of 0.8. The structure would eventually lead to or immediately led to a mapping of the epitope, and eventually led to an understanding of the neutralization mechanism. So from here, I'm going to jump to the other extreme of a very high resolution structure, that of AAV-DJ. Now, this is not new data, and it's entirely possible that we could do better now with improved hardware and software. But for a while, this structure held the record for the highest resolution EM protein structure until it was overtaken by apoferritin. We do get excellent maps assisted by the high 60-fold symmetry. And in terms of the refinement, we start here with a structure that we had earlier determined at 2.8 angstrom resolution. So this was a multi-step refinement where we started with torsion angle simulated annealing, did some remodeling, then gradient descent refinement. We added solvent waters conservatively, and 9 alternate rotamers, continued with gradient descent refinement. We then refined the effective resolution by using only the most ordered part of the structure, the core strands of the beta barrel, and refining this against the fifth order Butterworth parameters, we got a resolution of 1.44 angstrom. That's in fairly close agreement with the FSC that was 1.56. Overall, the correlation coefficient improved to 0.8. And we were also able to see many of the hydrogens. Now, oops, sorry. There we go. Now, this kind of surprised us because from crystallography, we would expect to need about 1.2 angstrom resolution to be resolving hydrogens or to start resolving hydrogens. And here at a bit worse than 1.5 angstrom, we're apparently seeing them. Now, we cross-checked this in the following way. We grouped all of the hydrogens into a single occupancy and then changed that occupancy and looked at how the correlation coefficient between calculated and observed maps varied as a function of this occupancy. And that's the curve here that I'm outlining here. We calibrated that to doing the same thing using side chain carbons, and that's this curve right here. So what we're seeing is that the hydrogens that we're modeling are about 1/4 of the expected strength as calibrated against side chain carbons. The middle curve here is for solvent water molecules. So they're not at full strength because we don't have full scattering at this resolution, because they're not fully resolved, and because of disorder. But there's certainly significant signal that gives us confidence that these hydrogens really are there. And indeed, we can find hydrogens for example, about half of the histidines in our structure, and that allows us to tell the protonation states. And what I can tell you is that we can also do pKa calculations, and in all 7 cases, we find that what we have observed by electron microscopy is what would be predicted by a pKa calculation. So we can also use this high resolution structure as a gold standard to go back and assess structures that we had obtained with earlier data and through earlier model refinements. And statistics are shown in this table here. We're looking at the RMS deviations in terms of atomic coordinates. Now, you'll see that in both cases, the accuracy is about 3 times better than the nominal resolution. And this is an effect that is similar to what we see in X-ray crystallography, but it is probably not as great. I think in X-ray crystallography, it's been estimated that the atomic accuracy can exceed resolution 5 to 8-fold. And here we're seeing it to be about 3-fold better. The remaining error is mostly in a small fraction of outliers. So in this row and this row here, we're removing, up to 10% of the worst differences. And we see that over half of the error is within the worst 10%. Now, I suspect that we see this as a big effect in electron microscopy because there's a greater variation between order and disorder in a typical EM structure than there is in a typical crystallographic structure. Now, also from this table, I was a bit disappointed that we didn't do better at 4.5 angstrom resolution. Now, the lower resolution structures were derived years ago, and it's possible that we would now do better with experience gained. However, I expect that we were too trusting of flexible fitting. That said, I would defend our approach in that we were using torsion angle simulated annealing, which is thought to be one of the more conservative ways of doing flexible fitting. But in retrospect, I think that at these types of resolutions, we would probably do better by rigid group refinement of an AlphaFold prediction. So yeah, I think I'll skip the illustrations at the bottom. The final example that I want to present today is illustrating our work on the AAV receptor. And we have long been interested in how AAV enters cells and traffics to the nucleus. And we've had a lot of false starts. After we determined the first crystal structure long ago, glycans were considered to be the entry receptors, but our research demoted them to the role of attachment factors. By this time, several other proteins had been implicated in the literature, but our structural studies made no progress, and we weren't able to establish their role through siRNA inhibition. And this led us to re-examine the whole question through genome-wide screening for cellular genes essential to viral transduction. So Nancy Meyer, who at that time was in my lab, teamed up with Sarika Pillay, a postdoc in the Stanford laboratory of Jan Karet. And the approach that they took was as follows. So Nancy took AAV and she pulled out the infectious DNA, replacing it with a gene encoding a fluorescent protein, and was able to show that you could then use fluorescence fax processing to separate out cells that were infected from those that were uninfected. Now, then Jan Karet's lab had a genome-wide library of mutants. And so they were able to run several rounds of selection, enriching for human cells that were able to avoid infection by AAV. So then with deep sequencing, they could identify the genes that had been mutated. There were about 40 that were important. The top ones they confirmed. And what I'm going to tell you about today was the very top scoring gene out of this screen. It was an uncharacterized protein at that time. And so we took the liberty of calling it the AAV receptor. It's a multi-domain membrane-attached protein. And Omar Dvolku, who was in my lab at that time, quickly succeeded in finding ways of expressing the business parts of this, the polycystin kidney disease domains in various different ways. And within a couple of years, we had structure. Our first structures were actually led by Nancy Meyer again. And so what Nancy saw was just a single domain bound to the viral surface. And this is a result that has been replicated by several different groups, all of us using constructs that had multiple receptor domains, and yet we're only seeing one of the domains bound to the viral surface. And so we switched to using cryo-electron tomography in collaboration with Scott Stagg's group at Florida State University. And they revealed that the proximal domain, this is PKD domain 2, domain 1 was pointing away from the virus in one of several different configurations, apparently not interacting with the virus. So this set up a couple of paradoxes because we already knew that deletion of this domain reduced transduction. Well, that could have been a direct effect, or possibly deletion could have been affecting domain 2. But then Nancy went on and found that if she added a solubilized PKD1 only construct, that would competitively inhibit transduction, which seems to indicate that PKD1 is directly involved in binding to the virus. So there was evidence for and against a direct interaction. And so we wanted to try to characterize this. My student, Ornella Weirakoon, then took on the task of surveying some other natural AAV variants to see if there was one that bound PKD1 more strongly. And eventually she was able to find one that bound about 50 times more strongly than the AAV2 virus that we had been studying. It's still a weak interaction, and we've never been able to get a complex of virus with just the PKD1 domain. But when we did a complex with both the PKD1 and 2 domains in our construct, we found some weak discontinuous density, unaccounted features that we hoped were PKD1, but the map was uninterpretable. We couldn't figure this out for months, and then we had some luck. I was playing with an AlphaFold 3 model of the receptor, searching for consistency with some prior cross-linking data. I found that by bending the interdomain linker by about 90 degrees, one of the beta strands of PKD1 passed through several of the stronger features of this disordered density. Then, with dihedral rotation of the domain, I found that I could rotate a second strand into some density, and then a third, and so on. And the final realization was that we were sitting on top of a symmetry axis. And when I added the symmetry-related copy that you see here, now we found that we were accounting for most of the strongest features of this disordered density. And it had been very difficult to interpret because what we were looking at were peaks where strands of different beta sheets were crossing at about 90 degrees. And so the peaks are where you've got two structures superimposed. Now, what's emerging here is a very good case for focus classification. However, we'd had several prior attempts at doing a focus reconstruction that had all failed. And so we proceeded with atomic refinement of this superimposed set of two domains and optimized those superimposed domains against the average reconstruction. I doubt whether we could have done this with some of the other refinement methods that were available. So, spoiler alert, I'm skipping a couple of steps to preview that we would ultimately be successful with a focus classification. But I want to show you the answer first. In green here, we've got the PKD2 domain that's similar to what we had seen in the complex with AAV2. And in pink here, we've got the domain that we'd never seen before. And this is coming out of a focused reconstruction of a spherical volume. But what was essential to know was where to place this, this mask. And I think probably our earlier attempts at focused reconstruction had failed because we didn't have the mask sufficiently well positioned. So the refinement was multi-step. I'm now filling in some of the intervening steps, and I'll go through them very quickly. We started with the by refining magnification against a homologous crystal structure, and this is much more typical now. We're seeing errors of well below 1% usually now. But we were surprised that when we put in the PKD2 from a homologous structure, now it's rotating by 10 degrees with a 2 angstrom shift. You can see with a pretty big improvement in the correlation coefficient. At this point, we started including the virus in our refinement, and the correlation coefficient goes up to nearly 0.9. We can now refine the resolution to be 2.7. This is by agreement with the atomic model. The FSC had told us it was about 2.6. This is a typical level of agreement that we see. And now we added the PKD1 from our mutated AlphaFold 3 model, where I'd rotated a domain by 90 degrees. On refinement, it moved a further 4 degrees, and the correlation coefficient improved nicely just for the PKD1. So combining it all, we're now having to refine against an unsharpened map because the PKD1 was not well resolved in a sharpened map. We got a pretty good improvement. We refined the individual B factors, restraining the differences between adjacent ones. If we then go and place our structure back in the sharpened map, we get a correlation coefficient of 0.83. So it was at this point that we'd managed to do the focused classification now that we knew where to put the mask. And so we pick up now with refinement into the focus region, the mask area. And so now what we're able to do is refinement of the receptor plus the neighboring bits of virus that happen to be within that spherical mask. And the correlation coefficient improves slightly to 0.81. The focused reconstruction is of slightly lower resolution. When we refined it, the resolution went down from 2.7 to 3.0 angstrom. So we're also able to refine the occupancy, and what will become relevant later is that for PKD1, this drops to about 0.5. Okay, so we end up after individual beta refinement with a correlation coefficient of about 0.9. How do we know that we got it right? So our mask doesn't incorporate any molecular detail. Here we're looking at detail within the focus reconstruction in cyan as part of the receptor. Clearly this is 3 angstrom-ish that we've been able to derive. This is for the other domain. Critical for us was what was going on in linking the 2 domains, and the linker is also pretty clear. To give you an overview sense, the PKD2 domain is in a similar position as we had seen in AAV2, except that it's moved about 2 angstrom. And what we're seeing afresh is this new domain here that we hadn't previously been able to see. Now, this is a very busy slide, and I'll talk you through it to illustrate the value of doing uh, uh, individual B factor refinement. And so our structures here are colored by B factor. For the viral surface, we're coloring between cyan and yellow. And so you can see that some of the most exposed loops have somewhat higher B factors on the virus, either green here or yellow down here. The receptor were tracing its chain with a cartoon tube that's colored by B factor, going from blue being the lowest to red being the highest. Now, I've also put in the focused map in brown. And so the first thing that you'll see from the B factors is that the very highest are coming from places where we don't have map. It's too disordered. And so this is what we would expect. The lowest B factors are blue here, and they are where the PKD2 domain is in most intimate contact with the viral surface. The B factors are restrained between neighboring atoms, but as you go from the N-terminal end here to the C-terminal end of the domain, the B factors are getting larger. And it's as if the interaction is stabilized at the surface here, but as you get further away, there is perhaps a larger and larger pivoting around the binding site. Now, if we go to the previously unseen PKD1, there is also a very small region in here you might or not be able to see of blue residues, where it's most closely associated with the virus, but then an even more noticeable decrease to higher disorder the further you get away from that site. You can see here we've got no interaction with the virus, and so we're assuming that, again, that this is pivoting around a bit. So, I think these are the sorts of things that you can learn with density functions that allow you to refine individual B factors. Now, I want to point out that we've got a rather extreme example of alternate conformers here, putting the results into context. So our samples were prepared at near saturating concentrations of the receptor, and occupancy refinement indicates that nearly all of the PKD2 sites are indeed occupied. So in cyan and pink is the structure that I showed you on an earlier slide. I'm now going to insert in a symmetry equivalent. This light blue corresponds to the cyan, and then this brown corresponds to the pink domain to which it's connected. And clearly, both the brown and pink domains clash. They can't both be at that site at the same time. And this is consistent with what we had seen in the average map of two domains superimposed. The occupancy refinement settled on miraculously 0.49 for each of these domains, and but only one of these can be in the configuration that's actually shown. So we've still got a missing one domain that we can't yet see, and we think that it's likely to be in a position similar to that seen in the complex of AAV2 by the cryo-electron tomography. And so we think that the other domain has to point away from the virus surface and is hanging off in one of several different variable configurations. So at this point, I want to wrap up and ask, what are we learning for doing a good job of refinement? And I think that we're learning that we should be able to do better with accurate calculation of atomic density, and that now this is fully tractable. We don't need approximations for expediency anymore. And that allows refinement of EM imaging parameters, individual B factors. We like to think, but it's unproven, that it leads to more accurate structures as we get the agreement between observed and calculated densities to be better. I would emphasize that this is a way that you can refine alternate conformers. We had an extreme example of overlapping domain structures, and clearly we can't expect atoms to be at their peak, at the peaks of an observed map. But this is also true if you were trying to refine side chain rotamers, uh, um, when you have complexes breaking symmetry, when you've got ligands that cause conformational changes, but the ligands are binding with only partial occupancy. In many cases, the parts of these structures that are different are too small to allow easy focused reconstructions. And so we think it better if you can be refining with methods that allow the components and neighboring atoms to be included. And so our goal is to complete the development of a module that others will be able to more easily incorporate in their own refinement packages. With that, I would like to thank a number of people that the recent work on this method has been built on earlier versions in which Eric Blanc, Andrei Korostolev, James Chen, Felsi Gabriel, Andrew Trzynka, Brynmore Chapman were all contributors. And then the examples that I've shown, some of the people most involved, Dustin McCraw on the antibody work, Qingzi on AAV DJ, along with Craig Yoshioka. And then on our receptor work, Omar Devolku, Nancy Meyer, and the recent work, Anara Wirakun. And I'd also like to acknowledge that much of the data for this was, for the examples, was collected either at PNCC or at Mizzou EM core. With that, thank you, and I'll try to address any questions.
All right, thank you so much, Michael. It looks like you've mostly stunned the audience into silence. But I did have a couple of questions. You've looked at a number of different serotypes of receptors and their complexes. Is there a pattern that emerges as to what type of map is the best to refine into, whether that's sharpened or unsharpened or enhanced in some way? What is your favorite?
Yes, we're usually using sharpened. We haven't experimented with the various enhanced forms. Particularly, we haven't looked at the machine learning enhanced forms. We do also look at the unsharpened map because sometimes there will be regions that clearly don't have the right sharpening factor that we can detect with the original map.
Can you tell us a little bit about kind of your workflow? So you've got a capsid map that you can presumably fit those atoms into really well. From there, how do you go to those images you were showing us?
Yeah, all of the images that I was showing were based upon just standard reconstructions. Apart from the earliest map of the antibody that was bound. For the antibody, for whatever reason, we didn't get strong enough map features to model into the native map, and we had to do a difference map calculation. And as Paul was emphasizing, when you're doing a difference map calculation, it's pretty important to get the scaling right. And weak signal can just disappear if you haven't got the scaling right. We notice this even more with work that I didn't show today, where we've got the binding of small ligands to the viral surface. And uh, we um, we have to be subtracting the right amount of uh, Coulombic potential uh, from the observed map. And that Coulombic potential has got to be calculated appropriately for the resolution of the atoms concerned. And so Paul was referring to some, I think, some reciprocal space methods of doing that. We're able to do it because we can refine the occupancies in real space, occupancies and B factors in real space, and we can subtract that density from our map in real space.
So I think in all of these, you've enforced icosahedral symmetry. Do you see much difference when you relax that?
Uh, Apart from our focused reconstruction, we haven't experimented other than with the cryo electron tomography. But in terms of SPA, we really haven't done it. And when we did the focused classification, effectively, we've got 30-fold symmetry rather than 60-fold. I say that. Yeah, I guess that's true. It's effectively 30-fold symmetry instead of 60.
And what software are you using to make your illustrations? And can you tell us what the different axes represent?
Oh, I'm using PyMol, and the axes that I was showing, I could maybe go back to one or two of them. Um, let's find one that's clear. The axes, this is a threefold axis of symmetry, icosahedral axis. This is a twofold, and this is a fivefold over here.
So you spoke about ensuring that the maps going into a difference map calculation are scaled properly. How are you doing that?
Yes, so we've got a sort of unorthodox way that we will um, using a model that we've refined against one of the maps. We will then scale each of the maps to that model, and that accounts for both a gain aspect and an offset that will be appropriate for the region that we're looking at, and according to the um, the refined B factors and so on that are all in the model.
All
So
right. I think, oh, I'm sorry.
no, go ahead.
I was just going to wrap things up. I think that covers all the questions. If you had something else, please feel free.
No, other than thank you very much, and I've enjoyed the day.
Thank you.